Terbinafine is another drug that works on the ergosterol synthesis pathway. It works by inhibiting squalene epoxidase, which converts squalene to squalene epoxide. Terbinafine is used to treat dermatophytoses, including onychomycosis or infection of the nail bed. These infections are notoriously hard to treat. Adverse effects of the drug include upset stomach, headache, hepatotoxicity, and taste disturbances. In my experience, these sort of non-specific adverse effects are not very high yield. If you have other high yield information down, like the mechanisms of action and the clinical use, then I would worry more about these toxicities, but try not to stress out about memorizing all of the non-specific toxicities. With that being said, do you remember how ketoconazole impacts testosterone synthesis? Remember, it inhibits its synthesis, and what side effects does it cause? Well, ketoconazole is associated with gynecomastia. Very good. So now this is an example of a unique side effect of a more commonly used medication that is more likely to be tested on than asking you about the taste disturbances associated with terbinafine. The next antifungal therapy we're going to talk about are the Echinocandins, caspofungin, micofungin, and antihelminthic. You can remember these drugs by thinking that candy is fun. These drugs work by inhibiting the synthesis of beta-glucan by inhibiting beta-1,3-glucan synthase. Without the synthesis of beta-glucans, the cell wall is incomplete and the fungi can't survive. This drug is especially used to treat this fungal infection. Can you tell me what fungi you see on this slide? This is a picture of Aspergillus fumigatus from a lung biopsy. And how can you tell? Remember, Aspergillus form acute angle branching, as we see here and we see here. Also, we can see the septate hyphae, for instance, right here and here. I know they're difficult to see on this image, but you can see the septate forming in the hyphae. Caspofungin is used to treat invasive aspergillosis as well as serious invasive candida infections. The next drug is Griseofulvin, a drug that targets microtubules and disrupts mitosis. Thinking back to cell biology, during what phase of mitosis are the homologous chromosomes separating to opposite poles of the dividing cell? That would be anaphase. Very good. So Griseofulvin essentially stops anaphase from happening and results in cell death. Griseofulvin is used clinically to treat cutaneous fungal infections or dermatophytoses. Do you remember what we'd call the ringworm infection or the dermatophytosis seen here? This would be tinea capitis because we can see that there is a fungal infection here on the cap or the head of this individual. The use of Griseofulvin is limited due to its teratogenic and carcinogenic potential. In addition, it can cause confusion as well as induce cytochrome P450 and increase the metabolism of certain drugs, including warfarin. Now time for a flash quiz. What is the mechanism of action of terbinafine? Terbinafine inhibits squalene epoxidase, blocking linosterol synthesis. Now we are going to briefly talk about the antiprotozoal drugs. These drugs are pretty low yield for board exams, so I'm not going to spend too much time reviewing them here. If you think back to your parasitology lectures, protozoa can be classified into causing disease in one of three areas. 1, the GI tract. 2, they can cause disease in the central nervous system. And 3, they can cause hematologic disease as well. First, let's talk about Giardia and Entamoeba histolytica. These two protozoa can cause disease in the GI tract and particularly cause bloody and non-bloody diarrhea. Do you remember which one of the two causes the bloody diarrhea? That would be the Entamoeba histolytica. Remember, Giardia causes a non-bloody, fatty type diarrhea. The medication used to treat Giardia and Entamoeba histolytica are metronidazole. Next, we have toxoplasmosis. Do you remember the triad of clinical findings in toxoplasmosis? That would be chorioretinitis, hydrocephalus, and intracranial calcifications. Very good. The first-line treatment for toxoplasmosis is sulfadiazine pyrimethamine. Trypanosoma brucei, we use suramin for the acute blood-borne stage, as well as Malarsarpro for the CNS penetration. This protozoa causes African sleeping sickness, so we can remember that we use Suramin and Melarsoprol by remembering melatonin sure helps me go to sleep. Trypanosoma cruzi causes Chagas disease. What are the two important clinical manifestations of a patient with Chagas disease? We would see dilated cardiomyopathy as well as megacolon. To treat Chagas disease, we can give nifurtimox. For leishmaniasis, we treat with sodium stibogluconate. In my experience, this material is pretty low yield, and in the entire Step 1 exam, you will most likely get 2 to 3 questions max on protozoa, meaning you will probably get only 1 question on the therapy. So please don't drive yourself crazy over this stuff. Scabies and lice are 2 pesky skin infections that can lead to severe itching in patients. They are highly contagious and transmitted directly by skin-to-skin contact. To appropriately treat and eradicate these infections, treatment must include the entire infected household or community to prevent reinfection. Here is an image of pesky scabies mite, and here is an image of a body louse. The most effective therapy for the treatment of scabies is permethrin, an insecticide. The mechanism of action of this drug is pretty cool. What it does is it binds to sodium channels that exist in the axon found in the mite and prevents them from closing. Therefore, the sodium present in the axon continues to leave the axon, resulting in a continuously depolarized axon that leads to a paralyzed mite. Malathion is another medication used to treat scabies and louse, and it is an acetylcholinesterase inhibitor. It has a similar mechanism of action in the fact that it continues to have acetylcholine in the neuromuscular junction, since the mite can't degrade the acetylcholine with malathion present, and that leads to a permanently depolarized muscle fiber and a paralyzed mite. Lindane, on the other hand, is a GABA channel blocker. So if you blocked GABA, would this result in a net excitatory or net inhibitory effect on the central nervous system? Well, we would actually see an overexcitation of the CNS because GABA is the inhibitory neurotransmitter. By blocking GABA channels, the lice or mites are overstimulated, leading to convulsions and death. I know, it's very morbid. Now we're going to discuss the mainstay of treatment against malaria. And do you remember what protozoal species causes malaria? That would be the Plasmodium species. Very good. The 4 major subtypes of the Plasmodium species is Falciparum, Vivax, Malaria, and Ovale. And remember, they are transmitted by the Anopheles mosquito. To learn the mechanism of action, we need to remember that the Plasmodium parasites spend part of their life cycle in the red blood cell shown here. In the red blood cell, the parasite breaks down hemoglobin into its constituent parts, globin and heme. The parasite takes the globin, degrades it into its essential amino acids, and then uses it for essential nutrients to survive. The heme is the toxic part of the hemoglobin molecule and must be handled by the Plasmodium species. In order to break down the heme group, it turns it into hemazoan, a non-toxic intermediate, and then stored away. The way chloroquine works is chloroquine prevents the conversion of heme to hemazoan, resulting in the buildup of heme, the toxic metabolite, leading to disruption of the cell membrane and parasite cell autodigestion and death. So remember, chloroquine inhibits the conversion of heme to hemazoan, causing a buildup of heme and ultimate parasite death. But it's important to remember that chloroquine is used only to treat Plasmodium species other than Falciparum. It's not used against Falciparum because Falciparum can develop resistance very easily to chloroquine. The way it develops resistance is because it has a membrane pump that decreases the concentration of the chloroquine drug. In order to treat Falciparum, we use tabacone proguanil preferentially. The side effects associated with chloroquine are retinopathy, as well as severe pruritus or itching, especially in dark-skinned individuals. In fact, the pruritus can be so severe that the patient will often stop taking the drug. Now let's go over the anti-helminthic therapies. Remember, the helminth are the parasitic worms that can cause disease. Questions like these on Step 1 are rather straightforward and are pretty low yield, so we're going to go through them pretty quickly. Nematodes include Enterobius, Ascaris, Strongyloides, Ancylostoma, or Necator species. The therapies used to treat these nematodes include the bendazoles. The bendazoles work by inhibiting microtubules in the worm. Parentyl pamoate can also be used for Enterobius and the two hookworms, but it is not used for Ascaris or Strongyloides. Parentyl works by paralyzing the helminths by it simultaneously releasing acetylcholine and blocking acetylcholinesterase. With too much acetylcholine at the neuromuscular junction, the muscle fiber remains depolarized and unable to repolarize, and therefore the muscle fiber is paralyzed. Tapeworms include the adult intestinal worm, Taenilia solium, Taenia saginata, Diphyllobothrium latum. The trematodes or flukes include Schistosoma, Clonorchis, and Paragonimimus, which are treated with praziquantel. I'll point out a few things here. For Taenia solium, praziquantel is generally used except in cases of neurocystic sarcosis, where you would preferentially use albendazole. For Echinococcus hydatid cysts, you would actually also use albendazole. However, at the end of the day, surgical excision of hydatid cyst is the only reliable treatment. Praziquantel works by increasing cell permeability to calcium, which causes paralysis. Careful not to confuse Parentyl with praziquantel because they look and sound the same, but definitely have different mechanisms of action. Other roundworms include Loa loa and Wucheria are treated with diethylcarbamazine. Ranunculus is treated with a physical extraction of the worm. The worm is tied to a stick that is slowly turned over time to remove the worm a few centimeters each day. These worms can be close to a meter long, and it can take a few weeks to remove. Remember, the extraction needs to be slow and steady so the worm is not broken and nothing is left behind. Onchocerca causes river blindness and is treated with ivermectin. Remember, ivermectin is also the drug of choice used for strongyloides. Finally, Toxicara is treated with albendazole or mebendazole. Now time for a flash quiz. What is the treatment of choice for scabies infections? And how does it work? What is the mechanism of action? The treatment of choice for scabies infections is permethrin. Permethrin binds and opens the sodium channel in axons, keeping it depolarized and paralyzing the mite. Now let's move on to the last major section of the antimicrobial chapter, the antiviral therapies. If we remember back to the virology chapter, what are the three major components of a virus? Well, we have the genetic material, 2, the protein capsid, 3, the lipid envelope. Very good. Another important concept about viruses that we talked about is that they rely heavily on human cellular machinery to reproduce, from using our ribosomes to translate their capsid proteins, to using our plasma membranes to make their lipid envelopes. This feature of viruses makes them very hard to treat and prevents us from using many of our antibiotic therapies. So why do you think that is? Well, remember, many of our antibiotic therapies target processes that are unique to bacteria. For example, we can target their cell wall formation, as targeted by the penicillin drugs, or we can target their unique ribosomal subunits, as with the aminoglycosides or macrolides, for example. So in order to treat viruses with drug therapy, scientists and clinicians had to find new targets that are specific to viruses, and also while avoiding the human cellular processes that are important for our human cells to survive. Basically, what all this means is that we as clinicians are unable to treat viral disease. The good news here, or the silver lining if you will, is that there's less material for you to know for step 1. So when we think about antiviral therapies, we basically can break it down into 2 main categories. Number 1, drugs that are used to treat HIV, and number 2, other antiviral therapies. We'll be spending most of our time talking about drugs used to treat HIV because that is where many of the breakthroughs for antiviral therapy has been. Outside of HIV, the only other viruses we can treat are hepatitis B and C, some herpesviruses, the respiratory syncytial virus, and the influenza virus. That's it. Oseltamivir and Zanamivir are 2 drugs used to treat the influenza virus. The influenza virus is a highly contagious and potentially fatal virus. Depending on the strain, the virus can cause serious and widespread disease. In the flu pandemic of 1918, 500 million people were infected with the flu virus. Even more scary, almost 100 million died from the virus. That was 3 to 5% of the world's population at the time. This virus needs to be taken very seriously. If we think back to virology, do we remember what family of virus the influenza virus is in? It is part of the orthomyxovirus family. It is a segmented, negative-sense, single-stranded RNA virus. Very good. Now, do we remember the virus's two clinically important surface proteins? Those are hemagglutinin and neuraminidase. Hemagglutinin binds to cell surface proteins of an epithelial cell and facilitates viral entry into the cell, while neuraminidase is important for the release of progeny viruses. We remember that by the mnemonic, hello and nice to meet you, to help us remember that hemagglutinin facilitates entry and neuraminidase facilitates exit. Oseltamivir and Zanamivir are neuraminidase inhibitors. By inhibiting neuraminidase, these drugs reduce the rate at which progeny viruses are released from infected cells. It is important to know that these medications do not do anything to kill the virus. Rather, they simply slow down the spread of the virus, which gives our immune system time to fight and remove the cells that are infected by the virus. It's important to know that these drugs can be used to treat both influenza A and influenza B, and they must be given within the first 48 hours of the infection. Do you remember the two most important symptoms of an influenza infection? That would be fever and systemic myalgias. Very good. Now we're going to discuss the antiviral drugs used to treat the herpes virus. These drugs listed here, Acyclovir, Famiciclovir, and Valacyclovir, have activity against varicella and herpes simplex viruses, but they do not have activity against cytomegalovirus. The drug Ganciclovir, which is discussed in other lectures, is active against CMV. These three drugs here, plus Ganciclovir, all have the same mechanism of action. These are all guanosine analogs that inhibit viral DNA replication by causing chain termination. Do you remember, is guanosine a purine or a pyrimidine? It is a purine along with adenosine. So you wouldn't be crazy if you asked me, well, guanosine is used in our own cells' DNA replication, so why don't these guanosine analog drugs kill our own dividing cells? Well, the reason they don't kill our own cells is because they are activated by kinases that are only found in cells infected by the virus. Our uninfected cells do not have the kinase, and therefore these drugs are not activated. So thinking back to enzymatic biochemistry, do you remember what type of reaction kinase enzymes catalyze? They phosphorylate molecules. Very good, they add phosphoryl groups. So, in infected cells, this thymidine kinase exists to convert the guanosine analog into a monophosphorylated guanosine analog. This monophosphorylated guanosine is then further phosphorylated into its triphosphorylated form, which is the active form of the molecule. These viruses are able to become resistant to these guanosine analogs by mutating the viral thymidine kinase and making it so the drug is never activated. It is important to remember that these drugs are only used to treat acute or reactivated forms of infection. These drugs by no means kill or eradicate the viral infection. They just reduce the severity of the reactivated oral or genital herpes breakout. It's important to remember that they are also effective against herpes simplex virus encephalitis. Unfortunately, these drugs can cause a crystalline nephropathy if the patient is not well hydrated, which can lead to an acute renal failure.